part of it. And this is because this is the local alignment, because always they use a local alignment, or because they look for the domain, and then they say this is the best result came from here, and this is really a false positive result, and that's why we lost the correct one, which is this one, by this, because here we are human, we try to play with the cutoff, and this is, we say this is the side effect of this method, the hammer. Of course, it's working fine, hammer search in some certain, but not always is fine. Depends about how you design it. So, and that's why we came with the machine learning method, which is just we try to do the way of the cross validation or the training in different way, because we have unbalanced data. Because if you remember in those classes last time, we don't have the same number of genes, because sometimes in this one we have maybe 2,000. This one we have 10,000, for example. This one, the poor guy here, this one type 6 here. Sometimes we have just only 100 genes. That means the data set is not balanced. And that's why if you have data set not balanced, then you have to be careful to have not overfitting, and also you want your model also learning also again. So that means we try to do nested cross-validation, which is help somehow the machine to learn more, but still it has side effects, because still if the data is small. Someone here will say, why not doing augmentation? Sometimes in the genome, the augmentation is not easy to do it. It will give you something really maybe more noisy than the current data set. But if you want to do augmentation by just enrich your data set, this will be, maybe it's not a good way, because we try it once, it's not working, and then we skip it, and then we say we will keep it unbalanced data, but we will do the way of nested cross validation. Anyway, but if you have balanced data, you don't need to do the nested cross validation. This is what they recommended actually always. So, and now this is our model. So the main idea about now, we have the genes, we have each genes. Okay, sorry. And now the genes here, right? We have these genes for one system, for example, it is Cas1, Cas2, whatever, Cas3, those are the name of the genes of the CRISPR. But there is two ways to encode it. Do we use the hard encoding, or we can use helping with the hammer model? So in the machine learning, if we encode it with 0101, when we try it, it gives us noisy with this system, with the CRISPR system. Maybe with the other system it will work out, but with the system, with the CRISPR system, we don't succeed by doing this and encoding each genome. Like, for example, we have 4 characters, right? We have A, T, C, G, and then you can say, okay, the position, for example, 0001 for A, and then for T, 0100, and then for C, you will have 0010, and then for G, you will have 0001. This is the easy way to encode. That means you will have here, this is the encode of A, and this is the encode of T, and this is the encode of C, and this is the encode of G. Right, you can do it in this way. So in this one, system is not working. However, this means not always this one, it will not work in the genes, because it depends from the system to the system. That means maybe the machine doesn't find a nice pattern to learn from this system, because this system, maybe they have special features, attribute that machine cannot recognize it. That's why we go back to the hammer model. But we try to do something in the hammer model. We don't use the normal hammer model, which I explained the last time to you just briefly. What we did actually, we take each subtype, and we already said last time when we look for subtype 1D, for example, here we said, yes, we take, let's say if we have subtype 1A, and 1A, it has like 6 genes, like Cas1, Cas2, Cas3. This is the cassette or the cascade. Remember when we say there's a cassette, we call those we call it a cassette or cascade. That's a set of genes next to each other, or loci. So, and then here, let's say we have maybe 10,000 protein or genes. So what we did actually, we first generate the similarity matrix with something, with some feature, with some condition, because there is a lot of way to generate a similarity matrix. And then after that, we do the MCL, which is the Markov clustering. And then Markov cluster gives us a set of clusters. Let's say we have cluster from 1 to the cluster 12. Let's say in this example. So now for each cluster we generate as we saw it last time. So that means if we have a set of genes, then we can generate, and we believe this is one family, then we can generate multi-sequence alignment. We use muscle, remember, and then from muscle we first example here we use HMM profile. We create HMM profile. That means for each cluster we'll have one corresponding HMM profile. So this is one version of the similarity. Then if we change the similarity again with different condition, we will have different type of cluster. That means, and then we change. We have three version of the similarity, which has taken into account some biology background and some biology observation about these proteins. For example, the length of protein we take it into account, the domains where they occur also we take it into account. I will not go to details. So that means for each subtype we have 4 versions of the Hammer models. So after that, what we did actually, then we just run the Hammer model. We take the all proteins from each genome, and then we run the all Hammer models. And then you can see it here that we have a set of protein cassette, and then run the Hammer model. Then you will have exactly sometimes you have value. This is the value came from the Hammer model, and this value is not the e-value. We take, this is the bit score. You remember in the BLAST, we say BLAST has 2 e-value, it has bit score or e-value. Also Hammer model, the profile, they give you also the same. They give you e-value, which is if it's small is better, bit score, if it's big is better. This is the main difference. We will have it in the lecture more details when we talk about the BLAST and the FAST algorithm. But this is here the bit score of the Hammer model. That means if it's big, that means this is good hit. If it's small, that means it. Sometimes so someone he told me it's good like 100. No, it reach sometimes 1,000, and sometimes you have 0. Like for example here, you don't have any hit. That means, and this here after that we actually provide this is to the matrix. It will tell us what is the cassette. This is each X is a cassette. That means the set of protein together. And in each column here we have the value from which Hammer model came, and we have the label because we trained this database in this data set which we came from that paper. We know the label of each of those, then we trained it here. So now we know the label. For example, maybe we call this is 1A, the label, and this is the second, maybe is 3C. This is the label, and this is maybe the label is let's say 2D, whatever. That means we know the labels. So then the machine learning learn this here and then try to do also a lot of hyperparameter optimization based on some parameter. I think everyone knows this. And then at the end it gives you the best performance. So in this paper we use not one type of machine learning, we use 3 types of machine learning. We use the classification of regression and decision, and then we have the extended decision tree, and then we have SVM. And you can see the performance here. And that's why here we have to be careful because we have to normalize the result because the data set is not balanced. And after that, this is here, we said this is the best one. And then we just want to show you more details about here. And you can see here each Hammer model. And then it will tell you, for example, and this is here the last thing we'll stop here. We said in the Hammer model, actually, if you have the same set of protein and you cluster this protein based to different cluster and you create Hammer model, you will have different result. For example, let's say this is type 1A. You can see with Hammer 1, we have 160 models, and in the type Hammer 2, we have, with this parameter, we have 160, but here we have one extra Hammer model, but here we have nothing. That means the configuration here is different, right? Because the Hammer model based in, if your cluster is good, then you will have result. If your cluster is bad, you will have bad result, and it affect your Hammer model. Because if your alignment is very similar, then your Hammer model, it will look for something similar. If your alignment has a lot of gaps, then it will give you a lot of noise. You have to be careful between how to select. And this is the way people try to work a lot in the Hammer model and try to observe these things happen. And they have to, when they work with Hammer model, they not like you should be experienced in your proteins, because each type of Hammer model, maybe the way we do it now, if I will apply the same thing in different family of the protein, for example, it will not work, because each protein has some feature and have some domains which is different than the other one. And at the end here, we said, okay, here you can see what is the performance of each method. This is the horizontal, and you can see here, this is the best one with the Hammer 4, the STR. With the card, the best one was the Hammer 2, but still this is the best one overall. Even like with the SVM, still this is the best one. This is read it horizontal, people, don't read it vertical, but just here. And then it will tell you this is also the best result with the Hammer 1. And then you can see easily that each different architecture of machine learning has extract different feature from different Hammer model. The same. Even now, someone will tell me, oh, these are different protein. No, they are the same protein. They are the same protein, but when you when you cluster them, maybe you have in the first configuration, for example, this configuration 1, this configuration 2, this is configuration 3, this is 2, for example, sorry, this is 3, and this is 4, and this is 5. Each configuration will give you different number of cluster, which you can see it here, they are different. And also, now the Hammer model, it will learn something different. For example, maybe in class 1, it has protein A and B, for example, and C with this configuration. But in the other configuration, maybe there is a protein A and B, they are not common in the same class, they are going to different class, they are separated. And that will affect your alignment, it will affect the Hammer model. It will, maybe it's good or bad, that's later we can see what the machine tell us. But anyway, so far, this is what we stopped. This is the main message about why sometimes Hammer model is not good. This is, I think everyone see it now. If you have question, just speak up, because I don't see you, right? I'm feeling that I'm talking to myself. Even I had lecture before with the undergraduate, the same thing. So now also, we want to provide also more evidence about this machine learning proposer tool, and then we select subtype or class 1D, and we want to see what is the important proteins. And then here it tells us here, for example, it tells us the important protein for this one, the tool, it tells you, because here we list the all type of all genes of CRISPR, but some of them very important to the Cas1D, some of them they are not belong to the Cas1D. For example, those here. You remember this figure? Again, just to remind you, just to see it here. Sorry, this one, for example, you can see type 1D, this is the set of protein which belong to type 1D, which is we have Cas6, Cas4, we have Cas10. Here the important one, we have Cas3, and then and there is another Cas which not belong to. But now we ask the machine, we give her the all protein, and we ask her which is the most important protein, and tell us Cas10 is the most important, and then it came Cas3. Here is most important. That means this is correct about what we published, and this is also again tell you Cas4 is also important, and this is the ranking. And then here they tell you this is not important for this subtype. But if we give her different subtype, it will give you different result. Sure. So after that, we want to see how also what's the parameter to decide here. No, this is based in the future they learn. They try to find here. Just they look for the future in the model, and then we extract this feature from the model, and then we plot it in the frequency to say how good the score is. So this one is related also to here, to the next figure. For example, here, the same thing, we ask build us the binary tree to tell us how the important proteins for type 1D. And then it says, if you have Cas10 in the beginning, if they find, like, for example, you remember the cassette, it came like this. And the order always is not fixed. So in this case, if this set of proteins, the cassette, or they start with the Cas10, then it's high, let's say this is, if you have cas 10, let's say the next after cas 10, it should be cas 3. That means this is because they learned from the model. And they said always, we have cas 10, and then after cas 10, it should become cas 3, and it will give you high. This is if true, because it asks if this cas 10, it said yes, then it will go here. And then it will see, okay, after cas 10, cas 3, say if I have always competition or composite of, or together combination of cas 10 and cas 3, then always it will be go to the cas group or subtype 1D. But sometimes we have cas 10, but it's not similar to the one in the training. That means it will think the model will say, it's not a cas 10, it will give you false. But again, she will ask, can the value in the middle, can we look for the value of cas 10 in the middle, not the high one? Yes. If the model say yes, means here we have yes, then he will find cas 3, and then we can tell you how these decision trees or extended decision tree define the correct one, which is actually, this is what happened in the middle. This is, we plotted from the training. The last thing we also was looking to it, which is we have some result, but not that correct, not very super nice. We try to have some, like, okay, which one here? This one? This slide? Say something, just say yes or no. Yeah, okay, fine. So now here, I want to see how important, or what is the important protein for the extended tree to decide which is the class. So from here, it will tell you, if she find cas 10, for example, for 1D, I ask her, what's the important 2 or 3 protein to tell me if this is this type 1D or this group 1D. This is the group. What's the important feature? And then that it will the tree here, it tell you, okay, if I found cas 10 with the high value, then I'll say this is the first feature, and then I will look for cas 3. If I found cas 3 with also high value in the score of the machine, then I will say this is definitely 1D. But if I have found cas 10 with the high value, and then I go here, I found cas 3 next, I see cas 3 but with weak value, this it should be not type 1D. Why? Because if you look here, cas 10 is not only occur in 1D, it's occur in different types. Occur here. That means many subtypes they have cas 1, but the machine learned that means for type 1D always should be a strong value for cas 10 and then cas 3, which is they next to each other. Because if you look to here, we have cas 10, but we don't have cas 3 at all in this combination. This is the model it learned. Because the score of the model, how the probability of when the model is learned, it gives you, for example, the prediction. This is the score of the prediction. And this is here in the extended tree, you can ask, give me the rule if what's the important feature or what's the important proteins to classify or to predict this belong to this group. And it will tell you this one. So this is the way, I mean, this is a function, because after you learn it, if your model is good, but if your model is bad, then you will have not a good result like this. This is now if your model is good. So now the other thing we want to do, since we learn from here, there is something connected or something associated. That means always to recognize cas type 1D, the most important is cas 10 strong and then cas 3 strong. Or sometimes we have also cas, if you go to here, for example, you have one case, is very interesting case, this one said, if you have cas 10 is weak, then you go here. And then you can see, sorry, that means it's not strong, because the machine has 3 different type of strong. Does the value always fix? No, it is learning, not fixed. It is learning. Sure. It is automatically where it's learning. This value we don't. That means there is not human in this, like not like a hammer. The machine it tell you what's the value, and then here you can see if this is not a very high value, then it go to the medium. If this is still not medium, go to the low. And for example, if it's here we have weak Cas10, then it doesn't take only 2 protein. It take more than 1 protein. That means if I have weak, yes, it's a weak Cas10. Then I said I need to look for Cas3. I found Cas3, but still weak. Then this is not enough evidence to say this is a group 1D. I need more. Then if I found Cas1, which is high, then I say, okay, this is now a good combination to tell this is bad, is a good bad to say this is a group 1D. So that means sometimes in one case, the machine tell you this is just 2 strong proteins is enough for me to identify the class. Sometimes strong result, strong hit, but sometimes if you have even weak hit, then I need 3 protein or 4. In this case, see, it has 3 proteins, 1, 2, 3. 2 of them is weak, but this, and then third one is strong. But if this is weak, then it will tell you the machine this is not Cas10. Sorry, not 1D. It's something different. Not 1D class is something different. So from this here, we think that there is something, we call it associated rule. There is something rule between these genes. And for each class, there is 1 tree. Yes. Actually, no, we take the average. It generates more than 1 trees. Actually, we submit all the result of the all possible, and then we take the average. Yes, it's not 1 class. Yes, yeah. For what? For the week, the machine we ask her, because after we do the prediction in the learning, we scale for each class. We do the scaling. Just sorted all the scores, and then we say this is from here. It's strong. This is medium. This is low. This is you can easy do it. Just for the model from each one, you can do it like this. This is the easy way you can do it for learning. To just see what is the correct one. Just put the all result, sort it, and then you can make which group is strong, which is medium, which is weak. So back to this one. Now by this, it gives us some hope that we have maybe there's something connection between this, which we want the machine learning tell us, or learning this associated rule. That means if I see some protein and the other one is missing, can predict the missing one, which we will do it with the regression here. Just as for straightforward regression. And now, for example, in some cases it working fine. For example, this one and this one. In some cases working bad. So what this table class? For example, if I have this Cas R, which is belong for 1A, and also we have sometimes Cas R also belong to 1D. They are same family, but they are working with different group of the subtypes or of the cluster of the proteins. So now this is our target. Then we ask the machine, if I have Cas R for is this the learning depends in the number of samples? Of course, if the sample is good, it will help. If the sample is small, it will not help. Sure. Yeah. So now we go to this one. What we want to see? We want the machine tell us, if I will give you Cas R, which belong to 1D, can you tell me what is the other protein which is more always, you see it in the training, they are connected to Cas R, which belong to 1D. It tells us, if you give me Cas R with 1D, always I say CS15 with this value of accuracy, and also I saw Cas5 with the 0.1, and also I saw Cas6. So for the subtype 1A, always the machine learning learn something that if this as a frequency or some rules, we want to make it as a rule, associated rule. That means If she saw Cas-R, always she saw those a combination of protein together. Cas-R for 1A. If she look for the Cas-R for 1D, 1E, it gives you different combination of protein. And those, it's not very exciting result, but it's promising if someone investigate more and more about those associated protein. Because why we want to do this? Because sometimes if I give you one protein, I want to see the all combination of the subtypes, and then if something missing, I already know there is something missing. And this, for example, here, let's assume the real scenario for 1A, it has Cas-R, and it has Cas CSA 5, sorry for the writing, and it has Cas 5, and it has Cas 6. So those are the combination of, let's assume for this cluster or this type of group of this, the subtype of this, the classification. So in the future, sometimes we don't have the complete genome, which is the complete genome of bacteria. It look always circle, but here we said, normally we said as just one line. Okay, we don't have complete, we have just snapshot sometimes, because it's sometimes when we do sequencing, we are not succeed to have the complete genome. But then I will have just, that means maybe this is the part of the genome, which is maybe we found it when we did the sequence, we found this part of the DNA of this bacteria. And this part of DNA has CRISPR system. But since it's just part, that means maybe the CRISPR system is broke down here. Just has those together in this area, because the remaining here, they are in this area, the remaining sequence, they are in this area. Those sequence, or those protein genes, they are in this area, but we are failed to sequence them. So now, I know this system is not complete, but if I ask the associated rule, I tell her, I will give you one of those. Could you tell me what's the missing protein here? It will tell me if the missing protein is Cas5 and the missing protein is SCA5. This is what we want to do, because we have a lot of data is not complete genome, and it has fragment or just part of the CRISPR system, not the complete subtype part of this. But we want to predict what is the missing one, why they are missing, if there's something damage in the system, or because the assembly, when the people in the lab sequencing this bacteria or this gene, they do some contamination. That means there's some error happen. It's some happen in the way they do the experiment. This would help us. This is what we want. So we were happy with it, with only 2. The remaining we are not very happy, and then we look to do further analysis in those 2 together. So this one now, yeah. So I did not understand this concept, missing one. So if you have, for example, this is the ideal way. If we have a complete genome, complete without, you have a bacteria, which is, you want, for example, for some lab, or you go once for some clinical lab, and then you sequence, take some sample, and then you find some type of bacteria. Or for example, from your mouth, you have a problem in your mouth or the stomach of anybody, you sequence some bacteria. Sometimes you succeed, you sequence the complete genome, which is, this is fine. Sometimes you could not sequence the complete genome because there is some error happen, you just sequence part of the genome. Let's assume you sequence this part of the genome. So normally, in the real scenario, the CRISPR system starting from here, in the complete genome. So now, but you sequence from here. That means you only cover this area in your DNA. So that means when you do identify the CRISPR system, you will find only 3 proteins, which is those, or 2 proteins. And 2 proteins are missing. Why? Because the sequencing was not correct. You lost this information. Right. So now, by having part of the complete class, let's say this class 1A, part of them, can I recover to see what is the missing proteins? That's what we want to do it here in the associate rule, to tell us how important and what's the missing proteins. This is the idea here. And this is what we call it now, the missing protein independence of the test. So this is we make the tool is learning from the data set, from the same data set what they learn this associate rule, and then we have now a new data set which has nothing related to what we trained. And you can see the accuracy is not that good. For example, this is the nice accuracy, but it still is 0.6. But this is if it's just one missing protein, this is the accuracy. But if it's in this case, for example, in this here, we have two missing protein, the accuracy is go to the, like I think 59, it's go down and down. So the tool is still struggling, it's not learning good to find the associating how they are connected this protein together. But if it's just one protein is missing, it works fine. It's not fine, at least is okay until 75% accuracy. But if you have two protein missing, it will be bad. And you can see also here, some hammer, this is hammer 3. You remember? This is hammer 3, it's working, the missing protein is working fine. But in the complete protein, you can see hammer 4 is working fine. Right? You remember this one? How this is good? See? In the complete set of proteins, with the same CRT, hammer 4 is working fine. But if in the missing protein, we have different hammer model working good. And see, that means each model has different features, and the machine learning different features from different model, even though that they are the same sequences. Anyway, so this is so far at that time we are happy, and then we say we will dig down, we will look in all again and again, but actually we get distracted with something, some project, and I think I will come back one day if some student interested in this area and this problem. I will come back to find, because this will help us a lot to solve many things. So this is here, and then we try to prove it. We went over the all databases, and we look for the non-complete DNA. That's fragment of DNA. It has CRISPR system, and we found two very nice cases. For example, this one and this one, which they have not complete. And our tool tells us the missing Cas protein is this one. This is what the tool suggests to us. Tell us this is missing Cas protein. And tell us for this, the other type, it tell us the missing Cas protein is this one. Then what we did actually, we try to look for what is the closer genome to the fragment DNA, and we find something complete genome closer to the one is not complete. And we found with the length of 1006 nucleotide distance, this protein is there. That means this protein is far away, but the tool can recognize this is the missing one, which is really very nice. Two cases, we are very happy with that. But we spend like more than 2 months, and then we find only 2 cases. At least we found something. In the beginning, it was really devastated, but at the end, we succeed to find really two proteins was really fine. And then we send it to the lab, they verified them, they are really the same protein, what's the tool found it, which we call it here positive. That means in the beginning we do it bioinformatic, we don't have any experiment in the lab to validate them. In that time when we publish this one, that's why we call it positive, because still maybe yes, maybe no, because a tool. And then we need someone to do it in the lab to verify if this is the correct one. So this is about the tool, and the last one we just do the comparison with the other tools, and you can see the performance of the tool is fine. So far this is about what we remaining from the last lecture. Do you have any question? Can you say anything? Do you have any question? So no? Okay. You want to say something? Okay. No. I hope you are with me, not like I'm talking to myself. Yeah, so today we'll talk about a new lecture, which is, you remember in the multi-sequence alignment, we look for the phylogenetic tree, how the phylo tree is building. Today we'll talk about how we build the phylo tree, what's the concept, what's the way we find behind this phylo tree. Okay, so what is the motivation? Let's start by the motivation. Can you say yes or no if you are with me? Just even write it down in the chat. Then I just or just put thumb up. Just
Yes.
okay. Thank you. Yes.
Yes, with you.
Yeah. Thank you. So now we'll talk about phyla trees, and we'll talk about how important how no we know how the important, but we will go deeper to see how we construct the phyla trees and what's the motivation behind this. So the motivation behind the phyla trees or phylogenetic tree, some people they call it phylogenetic, or some of them they call it phylum, or some of them they call it phylogenetic trees. This is everyone really look at it, and they try to use it. But the majority of the people, when they hear this, the name, or sorry, when they hear the name, always they think about the evolution. That means always phylogenetic tree, I will look for the evolution, which actually is not true. Not always the phylogenetic tree you will look for the evolution. There is a lot of things we can do with the what could you do? Okay, now it's fine. Okay, sorry. Yes. So So many people, when they hear about phylogenetic tree or phyla trees, they think this is just about the evaluation. On one hand, is yes, people they try to do the evaluation using this one, but also it can use for many things. For example, we can use it for trace back the history of the two genome to see how they are, or two genes from different bacterium that we can trace back the history, or we can also look for the relation between them in the way, for example, how they are closer to each other and how they are similar, just not the relation, even the similarity between them, how they are close, are they are far away, why they are why in the genome location, why they are far away, would they have something in common between them, or if there is something happening in the genome destroy this relation. So far those here. Now even the phylogenetic tree, they use it also in the study of the language, how they're evolving the language from one language to go to different language. In the next slide, I'll show you the example. So normally in the biological or in the biology, always if they say phylogenetic tree, we have two, always people look for two things. Either they use the example of organism, like for example, we have human, monkey, dogs, fish. This is the famous architecture, or the famous way how what they draw it, and they tell you this is how the distance. Now, be careful. You can see this is very close, but you know what is the distance between from here to here? It's not only for multi-sequence alignment. I will come to that. It's not always for multi-sequence. It is, we can use it for multi-sequence alignment, or we can use it without multi-sequence alignment. You are right. We'll see it in this lecture today. Yes. So if you look to here, for example, of course, from multi-sequence alignment, you can generate a phylogenetic tree as we did it in the practice session last week. But also when we construct it, we can construct it without multi-sequence alignment. We use distance metrics, for example. We'll see it in the next slides when we construct this phylogenetic tree or the tree. So if you look to here, don't scare. They say, oh, human and monkey, they prove it, they are very close. You know, this what's the distance here? They say this is more than 1,000 millions years to they will take monkey to go to the human, which is really something. Just they put it as theoretically. That means just to say, we prove it, and there is just this number. You to say to the evaluation or the way monkey go to the human, it take him more than 1,000 billions years, which is something ridiculous for me, because I'm not famous with the evaluation. I'm famous in the evaluation of the bacteria, not in the big cells. In the small cells, yes, but not in the extreme cells or big cells. So this is one example. The second example, also, people they say, no, we are not going to use the organism. We will use the sequence, which is any sequence, other protein, DNA, RNA. And now the question here, for example, for us, this is important now for us, the question is, which sequence I can use? Do we have really some specific sequence I can use, which is give me more information or more accurate related of this if I want to study a set of organisms. Let's assume we have a set of organisms. This is, I cannot use the bin here. Can I use it in different way? Let's assume where is the, no, there is, I cannot use the bin here. Because, yeah, I want to write something, sorry. I will make it here, enlarged like this one. So let's assume we have from bacteria, oh, no, I need to, let's assume we have from bacteria 1 to bacteria N. And I want to study those bacteria. I'm interested in this bacteria. I want to do some bioinformatic analysis to study those, how they are close to each other, how if there is something happening in the genome wise. So can I use, I have more than one option. Maybe I will use DNA to study them, or I will use the protein, or RNA, and then I'll use the protein, right? To have something here, if I will apply the phylotree or phylogenetic trees, right? So now, which one is the good one? So there, if you want to study generally, in general, if you want to do in generally, it's better to use something, call it, okay. We can use something, we call it 16 sRNA, which is the ribosome RNA. 16 sRNA. 16 sRNA. This is N, sorry. The 16 RNA. This is the ribosome RNA, which is the most conserved RNA. This is not a protein, it's not a DNA, it's a RNA, this is the main RNA concern or play a major role in the center dogma. Remember, we have a DNA, and then we have RNA, and then we have the protein. Who doing in this step? This is the 16 ribosome RNA. So we have two types of the ribosome RNA. We have the 16, the big one, and we have the 18 sRNA. So here, if you want to look in the genome-wise comparison between those bacteria, to see how they are close to each other in the genome-wise, instead of looking for the whole genome, it represents each RNA, 16 RNA has represent one bacteria, and another 16 RNA represent another bacteria. It came from that bacteria. That each bacteria has 16 sRNA. And this is not, also keep in mind, this is not one version of 16 sRNA. Sometimes in one bacteria you have 10, 16 sRNA. Not 8 of them, they're exactly the same, and 2 of them is different. But still, this is the most conserved RNA in the bacteria or in the archaea, if you want to study the complete genomes. But if I want, for example, myself, I want to study a set of genes, for example, CrisprCas genes, then I will not use 16 sRNA. I will use the CRISPR system, and I will build my phylogenetic tree like this. This is my trees, based on a set of genes. This is also phyla tree. This is our phylogenetic trees. Those here, I compare this set of genes with the other set of genes, and then I build such tree. So that's based about what you want to do. Well, for example, here I use a protein. I don't use the RNA. So that's also, you have to be careful when you do analysis in the future. If you want complete genome, that's complete genome to genome, then the best way, the 16 RNA. If no, you are looking for a certain proteins in the gene, for example, from bacteria 1, I look for just a certain protein, which is the CRISPR protein. Then from bacteria 2, I look for the CRISPR protein, which belong to the bacteria 2, and so on and so forth. Then I don't need to use this one because it's not related. I just want to look for those proteins, how they are involved to each other, how they are related, how they are close. Then I will do this one, right? This is here just to tell you. But if someone tell you why 16 RNA, because it's more, there is less mutation, is more conserved, and it's always for millions of years in the bacteria, it keeps the same without any mutation. That means the area which generates the 16 RNA in the genome, it is really a conserved area. And also it is a core genes, because you have to remember in the bacteria, we have something we call it essential or the core genes or RNA, and we have non-core RNA or no-core. That means something is not not essential for the cell, and something is really essential for the cell. 16 RNA is the core for that one. Anyway, so I think this is clear now to see what is why just we can use more than one different version of input sequence for generating such tree based on what's your task. So now here you can see an example about the language. People, they use the same phyla tree or phylogenetic tree to study the language of, for example, in Europe. For example, in Indo-Europe language tree, they do it with the same thing with the phylogenetic tree. They want to study how this language evolved during years, and from where they came, how they are similar to each other. For example, here, if you look to this part, this is here, it came always the Latin, and you can see here you have more Italian, because Italian is very close to the Latin, but if you go a little bit up, you'll find the Spanish and also the French and so on and so forth. And this other group of language which you will have came, the English came, the Dutch came, the German came, because they are also came close to each other. They have really close to each other. And then if you go a little bit down, then you can see the Swedish language came, or from the Scandinavian country they came. This is another type of language, and they do it with the same way when you generate the phyla for the DNA. So, and also here they can see here the Greek language. Here you can see it in down how they can. And in this one study is funny thing. If like for example here you can see the Lithuanian language, the Russian language, they came from different also, and also is linking for the what is the Finnish language. But anyway, the surprising thing here in this study, they prove that Hungarian language is not part of the Europe language. And even the people in Hungary, they were really very angry that time when they published this. It was even in the media. That means. And they prove it. They say it's not belong here. It's not when they do it. So the question, how they build this one, how they use phylogenetic tree to build this. The easy question is if you have the similarity, or if you have the distance matrix or similarity matrix, you can build a phylogenetic tree. You don't need alignment. You need just some similarity, and you have language, and you create a similarity between language based on some criteria. That means we already know, that means we have some, not as a word, that means because in one language, how many, like for example, one language, let's say one genome. Let's say one language is one genome. It has a lot of genes, it has a lot of RNA, it has a lot of proteins. Then you can do the same thing. You try to measure a way to do a similarity or distance, and then you can do here. And the structure here, what do you mean structure and history? Maybe yes, for example, but they take it also into account the structure history, because they said even this here is present the distance, how they are close to each other, or how they are, maybe they are in this one, they say they are common language. Like for example, if I take this point here, I hope you see my or let's say I take this one here. If I am in this node, I'll say in 6,000 years or a million years, those all language is one. And then after the time, they try to invent a new language, which is, this is the Irish people go up, and this is here, it came the Latin here, and the other language, the French and the Spain, and then it go down, it came the English and the German and the Dutch, and together. Yes, this is the same thing as the similarity or the distance between the organism, or also between the sequence. So the main message here, that means it's not always phylogenetic tree based. We use it for the biology. Even you can use it for different purpose. But the main thing, you have to be what? you should have a criteria to create a similarity matrix for that. Anyway, so now for bioinformatic, actually, we use it, for example, for multi-sequence alignment. Yes, one thing we already saw it last time. Or we can use species tree, or we can also look for finding disease-related genes in the family. You can use it also to study, not always evaluation, just not like you look for the evaluation always, not always evaluation. They have many things you can study with this phylogenetic tree. Also, you can do the clustering, and also you can use gene expression clustering. So that means those all application for the phyla tree or phylogenetic tree in the bioinformatic. If they have accept this tree language, then we should we should what? We should also accept the human tree. No. Okay, no. Because biology is not as a car, it's not as a tongue. Biology is different. There's a lot of exception in the biology. Can you tell me in biology, I can prove it, there is always in anything, even in the single cell, you have a lot of what? You have a lot of mutation, you have a lot of exception. This is not the right thing. There's a lot of exception. That's really, and this is not. And this is here, you can build it because this is something they can trace it until like what? 200 years. But this is here, if you look here, can you go back 1,000 billion trees and they say they can process? No, this is they can easily traceable. But this cannot be easily traceable. There is no way to trace this one. This is just hypothesis. It's just theory. Just theory. It's hypothesis. No. Why we can't test with CRISPR? How you can trace it with CRISPR? CRISPR is adaptive immune system. It's not evolution. It's adaptive. Even we call it adaptive immune system. They don't call it evolution system. Why they call it adaptive? Because it can be just adaptive based on the situation. For example, if you are in the desert.
Yeah, doctor, can you hear me?
Sorry. Yes.
Yeah. Yeah. With the help of CRISPR, as you know, the researchers are bringing back the woolly mammoth, which was 10,000 years before in Russia. With the help of CRISPR, you can compare the whole genome. Okay, so that you have find the
you will not
20 mutation between the monkey and human. You can do these mutation in the
no. You misunderstood. Yeah. You misunderstood even the concept of the CRISPR gene editing. You say you already published one paper.
I did my thesis. I did my thesis on CRISPR-Cas9 and I derived the mutation.
Yeah, but mutation for how many mutations you deliver? In human, if you have one mutation, the human he will die. If you have just one mutation in the human, he will die. One mutation in human, he will die.
Oh,
in the human.
I inserted.
And human, if you just go in one cell.
Genome, I inserted the 7,000.
I can do one thing. I can, Muhammad, I can do one thing. I can challenge you in your presentation. I will challenge your professor as well. Even your professor, I will challenge him as well in this concept. Let's do it in your presentation.
Yeah.
For the defense. Yeah. I can challenge him. Yes, okay. And I will prove you and him. If he's agree with you, I will prove this is wrong. That's because I don't want to go to details because people here they are not aware about this concept more than. But I will challenge. Just remind me about this topic.
Okay, but not during the defense.
No, not during the after. No, of course that's not. Yeah. The defense is done. No, after because after we gather together, just I will challenge that. That's question.
Okay.
Yeah, and I will give you, what? People they will give you Nobel Prize if you prove it. If not but anyway, so
no.
The language is the main message. The language it can be easily trusted because it's like 200 years ago we can trust it back. It's like 500 years ago we can trust it back. Even to 600, 3,000, even 1,000, 2,000 we can trust it. But more than that you will not. Anyway, so. Now here we say the main concept here, what is the bioinformatic message here for us, or what we do with the phylogenetic tree or the the final tree, we can use it for multi-sequence alignment. We saw it in the example, or we use it in different ways. So the interesting thing here, you can even look for the clustering and also gene expression. This is a nice example I put it to you for the cancer here. Gastric cancer, for example. Here we have, for example, we have 93 patients in this figure, and we have 3,900, let's say almost 5,000 genes expression on this gene expression to the cancer. So those are in the row here. Those are the number of genes. Each row is one genes, right here. And now you can see here in this figure, we have two things. We have the clustering, and also we have also phylogenetic tree. And now we can see we have two different clusters. That means group 1 of the cancer, for example, and this is group 2. And also the gene expression related to the cancer. And you can see here clearly in this area is highly expressed, because you can see this is the heat map. It tell you how it say it's highly expressed or not. This is the scaling. So in this area is very highly expressed for this group of the cancer. And this area, it's highly not gene expressed for the other cancer. And in the middle, you can see there is something what mixed up. But even if you go here, for example, let us say I go here. In this one also, you can see there is clear highly expressed in some here area. But if I go to the other corner, that means those genes in this one here, they are very highly expressed for the certain cancer, but the other one is not. It's not very highly expressed here. And this is, it gives you two view. For example, the correlation between the gene expression within the clustering, and also the distance between these genes together within related to the cancer. Right. So this is how to interpret such thing. And this is also cool things we do it. Even if we go back to see with the, for example, with the finding disease related to the genes in the family trees. Also, this can help us a lot to do these things. Okay. So now this is just, so now one of the problem here. So can we have evolution by the phylogenetic tree? No, we don't have always the evolution by phylogenetic tree, because especially in bacteria, let's talk about the bacteria is easy. I'm not going to the human for the bacteria. We don't have always have the evolution, or we have the correct trees, or the species of the trees not always came correctly as we assumption, because we assume, or people when they say generating this phylogenetic tree, they assume two things. They say always we have ancestor, or we have a father, and then we have also the father came the children or the generation, and those generation they also generate another generation, or they evolve without corresponding to the father. Yes, and this is the way. That means each one can be involved. Means here we said involved means he can eating, nutrition, sitting in different country, have some disease. That means it's not that always the father he will have the same thing. That means those are the involved. In the bacteria also, for example, if the bacteria living in one area, after it's generating two bacteria, they are not connected to each other, because they have maybe they will still living in the same area, or they are splitting different, they go in different way. So this one, it gives us really a problem if we use this assumption, because normally in the bacteria we have something we call it horizontal gene transform. That means this bacteria, which is from type, let's say X, which is far away from the other bacteria, which from type A, like for example, this is type A and this type X, they are far away, they are not close to each other. Even you can see here, we try to illustrate this in the figure by saying this is bigger and has some more tails, this is just small one. They also can be connected to each other, and they also can be changing or transforming or changing some genetic information. That means maybe some genetic information came from here to here, or the other way around from here to here. So that means this, it will affect your phylogenetic tree, because principle they should be far away from each other if you compare them, but because they change some DNA, then they will look in the trees here. If you look to this example now, they will look here, they are very close. For example, this type of archaea, this is what people propose in the phylogenetic tree. They say, this is here our ancestor here. They say just one virus or one bacteria, and we came up from all the human all anything living in the planet, they came from one single cell. This is the theory. This is always the evolution people, they do this theory, and they say human goes here, and this is the archaea here, and this is the bacteria here. So this is they use it with the 16S RNA, just to have it like that. Okay, so now we know those are, let's assume those are group far away from this group, but if there's some happen in the horizontal gene transform, they are changing some genetic information. That means they are similar, and that means they give you a wrong idea. So this one thing. Also, this is cyanobacteria. Also, it's also involved in some high cell. For example, in the sea or in the ocean. Also, they some protein is of the cyanobacteria involved in some cells of the, what do you call that in English? I don't remember. الطحال او الشعب المرجانيه، ايش نقولها بالانجليزي؟ اللي حد عرفها. Anyway, okay, I say it in Arabic, sorry. Anyway, so now you can see that when you sequence, they say, oh, they are very close, but they are not very close, but because there is something here have been the horizontal gene transform happen between those, and that's why this one, okay, of course, thank you. That's what happened also, because cyanobacteria also in the sea is always there in the ocean and always transfer some genetic information from that. Thank you very much. So that's here now. It will tell us now how those information, that means it will give you wrong trees. That means not always your phylogenetic tree it will be correct, because this is the problem. It happens sometimes when they transform some genetic information, right? And that's why you have to be aware always. And this is the way they will transform the horizontal gene transform. They call it the conjunction. This is between all of bacteria and archaea. They do it very long, every frequency, very frequency. I would say very frequency. So this is just keep in mind. So now the question: what is the basic setting? Yes. Yes. Exactly. Yes. So now, if we say what is the basic setting of this phylogenetic tree, we have a basic setting which we call it first. We don't call it node here. We call it taxa. The taxa is here because we talk about biology. In the biology, we call it taxa. Like means each character or each set of character. That means here is one taxa. I think. Oh, we have to stop, right? It's almost 1 hour, right? Or we already take more than 1 hour. So, okay, then I will stop here. So I will stop here. Yeah, I will stop here, and then we can, let me just stop here, and then